This is the second morning of our main experiment, and we're leaving early in the morning because we need to do our experiment at low tide when the water flows from the main One Tree Island lagoon. There's Kenny smiling in the morning. We need to do it early in the morning when the water flows from this main One Tree Island lagoon to a smaller lagoon over a shallow reef area and then an algal reef flat.
Yeah, but it cycles through the day, right? So that morning, but each day passes will go later and later on. Right?
So this shows some of what the water looks like in the lagoon behind our study area. And you can see there's a lot of algae and some coral there. And here's our study site and the boat that took us there. So that's to the left there is the algal flat area.
So we have taken the GPS coordinates of our stations. We have got 3 transects and experimental sites and 2 control sites where we're not changing anything. And we have points up at the front of the main lagoon, one in the middle at the beginning of the algae flat, and one at the end of the algae flat, so we can get the metabolism for the algae part and for the coral part separately.
And so you can see there this the front. The main thing is the lagoon out in the center of the screen, and then comes this very shallow coral zone that we're studying, and then in the very much the lower part of the screen is some of the algal flat area. And so we're panning across our control stations and over to our experimental site.
Right. But those algae are not regularly part of the reef. It's turf algae just are stuck on the reef. Most under beneath that you can see a bunch of corals, small corals between the grooves.
At the back of the algal flats, every day we're taking these dye tracer samples. Here I am taking some tracer samples for the day, and so we inject some dye into the flow, and it lets and we measure a transect across the back. It lets us see the direction of the flow, the speed, and how much it diffuses as it goes across the wall.
Okay, here we sample for the biochemical analysis that we do. It's the back side of the station. Those are like small pike in the pie crust. It's not the ideal sample site because it's difficult to embed the bottles underwater.
So we're measuring mostly alkalinity, DIC, and pH, and the dye tracer, but in some stations we also measure oxygen and nutrients. It's a DIC bottle that Kenny's filling right now. And that will get brought back to the United States probably for measurement. And most of the other items we measure at a laboratory on One Tree Island.
Another view of the algae flats and the coral zone in the front. We see the enrichment site, the experimental site here, and now we're switching over to one of the control sites, and another one is following further down. So by comparing the control sites with the experimental sites, we see how much enriching the water with additional alkalinity enhances the growth of the corals.
And you can see the water flowing down from the upper lagoon to the lower lagoon across our study site.
And that is our video diary for today.